Today we will be reading about the macular dystrophies from fourth edition of Yanoff and Duker. The, what are macular dystrophies? The process of premature retinal cell aging and cell death, generally confined to the macula, in which no clear, demonstrable extrinsic cause is evident, and a heritable, genetically determined enzymic defect is confirmed or implicated, is macular dystrophy. The key features of these macular dystrophies are yellowish material within or beneath the RPE. Next is loss of macular photoreceptors and the RPE cells and the loss of central vision. What associated features? Retinal pigment epithelial and choroidal atrophy commonly limited to the macula. Bullseye appearance seen rarely. Pigment lumps in the posterior pole, mid-peripheral or far peripheral seen rarely. Optic atrophy, retinal vascular attenuation, macular edema, and choroidal neovascularization seen rarely. Introduction to macular dystrophy. The macula is the center of all the human vision and is critically unique to our visual function. It is irreducibly complex design, requires many unique proteins that allow the light to be converted to the neuronal impulses, that is phototransduction. These orders, while rare, are important not only for the individual affected but also because they allow a window into a better understanding of the macular function in general. Macular dystrophies allow us to see the clinical abnormalities of specific gene defects in the complex systems involved in the macular function. Molecular genetics has been instrumental in unlocking the secrets of these mechanisms and has allowed us to peer through the keyhole for a better understanding of the macula. The first retinal degeneration marked by the genetic linkage was one type of X-linked retinitis pigmentosa in 1984. Subsequently, the linkage of autosomal dominant retinitis pigmentosa to chromosome 3 was achieved by the Humphreys et al. This allowed Dreja and colleagues in 1992 to discover the first rhodopsin mutant associated with the disease. The first macular dystrophy to be genetically linked was North Carolina macular dystrophy in 1990 by Small et al. Since then, there has been so many important contributions by many groups around the world that even cataloging them is a formidable task. Fortunately, we can refer the reader to the online version of Macular Classic, that is the Mendelian Inheritance of Man, which is now available only online. Other helpful online resources include RecNet and the Retina International. From the clinical perspective, significant phenotypic variations associated with any inherited disease are the rule. For example, differences in the phenotype can represent mutation in different genes, different mutations in the same gene, and or variability in the genetic background in which the gene is expressed. The clinical phenotype can be further modulated by the environment. Collectively, these factors may make it difficult for even a skilled clinician to make the correct diagnosis by the ophthalmoscopic alone. Interestingly, many of the most common macular dystrophies are marked by the deposition of yellow subretinal deposits, leading to the cellular dysfunction and death. However, pathogenesis of each disease is unique. The table demonstrates how identification of the genetic phenotypes has given us insights into the macular dystrophies. The other table categorizes the macular dystrophy by the location of the defective cell. So, the first table, which is examples of cellular specificity of the macular dystrophic candidate genes. First is RP specific, according to the cellular location RP specific. What are the RP specific means? First is the autosomal dominant based disease, which is linked to the chromosome 11 long arm 12.3, and the gene is VMD2 and the bestrophin, and the bestrophin is a chloride channel. Next is autosomal dominant Sorsby macular dystrophy, chromosome linked is 22q 12.3, and the gene is TIMP3, that is a tissue inhibitor of metalloproteinase. The next in the RP specific is Malaysia, Malattia, Malaysia levantinase, that is also known as Downey honeycomb macular dystrophy, linked to chromosome 2p16, and the gene involved is EFEMP1. The gene is EFEMP1, which is the EGF containing fibrillin like extracellular matrix protein 1. When we come to the rod-specific retinal dystrophies, autosomal dominant Stargardt's macular dystrophy linked to chromosome 6q14, and the gene involved is ELOVL4, that photoreceptor specific elongation of very long chain fatty acids. Next, the cone-specific retinal dystrophy is autosomal dominant cone dystrophy linked to chromosome 6 short arm 21.1, and gene involved is GUCA1A, and the full form is guanylate cyclase activator 1A. Next, the conrod specific. The first one is autosomal recessive Stargardt's linked to chromosome 1p22.1, and the gene involved is ABC4, which is the ATP binding cassette protein found in rods and the foveal cones. The next in the conrod specific is autosomal dominant adult foveal macular dystrophy linked to chromosome 6p21.2, and the gene involved is RDS or peripherin. And where is this gene? Cone and rod outer segment glycoprotein. Cone and rod outer segment glycoprotein in the disc membranes for the structural integrity. The purpose of this chapter is to highlight the clinical features of these macular dystrophies, discuss the pertinent molecular genetics and the molecular biology of these diseases, and correlate the functional consequences of the mutant gene products in the framework of the known anatomy and physiology of the retina and the RPE. Obviously, every disease cannot be reviewed. However, the intention is to convey certain underlying biological principles that can be extended to understand the molecular pathogenesis of the other disease processes. With the advent of gene therapy, stem cell therapy, artificial vision, and a plethora of new retinal pharmacologics, proven therapy for these disorders is in the near future. In addition, therapies to halt the progression of the CNV have progressed and can benefit some who develop this complication. The other table was macular dystrophies and the example genes, like the Stargardt's disease. Stargardt's disease, if inherited in an autosomal recessive fashion, is linked to chromosome 1p22.1, and the genes involved are either ABCR or the ABCR4, that is the ATP binding cassette transporter in rod and cone outer segments. Next is the Stargardt's disease butterfly pattern dystrophy, which is autosomal dominant, is linked to the chromosome 6q14, also known as STGD3, and the gene involved is ELOVL4, photoreceptor specific membrane bound protein for the elongation of very long chain fatty acid. Next we come to this disease, inheritance pattern autosomal dominant, and chromosome linked is 11q12.3, also known as VMD2, and the gene involved is bestrophin gene, which is responsible for the chloride channel. Next we come to adult vitelliform outer dystrophy, which is autosomal dominant and is linked to chromosome 6p, and the gene involved is peripherin or RDS, and this is responsible for cone and rod segment glycoprotein in the disc membranes for the structural integrity. Next dystrophy is Sorsby macular dystrophy, which is autosomal dominant and linked to the chromosome 22q12.3 and is responsible for the gene TIMP3, tissue inhibitor of metalloproteinases. Next we come to Malaysia levantianus, that is also known as Doyne's dystrophy, which is autosomal dominant linked to chromosome 2p16, and the gene involved is EFEMP1, that is the EGF containing fibrillin like extracellular matrix protein 1. Next is North Carolina macular dystrophy, which is autosomal dominant and linked to chromosome 6q and also known as MCDR1, but the gene is not identified. Next is cone dystrophy, which can be autosomal dominant, autosomal recessive, mitochondrial, or X-linked. Multiple genes are involved, and it's also known as CORD, cone outer retinal dystrophy, 1 to 8 subtypes, and multiple chromosomes are involved. So next we will discuss the next part about the most common macular dystrophies, Stargardt's disease, and the fungus flavimaculatus.